I'm originally from Turkey. I did my master's and PhD in Zurich, and then came and started working with Andres 3 years ago. And today I will be talking to you about our recent paper called Matrix Producing Neutrophils Populate and Shield the Skin. But before I do that, I wanted to make 2 points. The first one being that this project has been a massive effort. If you look at the paper, you'll see there are 3 co-first authors and 3 co-senior authors and many other authors on it. So it's their work as much as mine, and it wouldn't have been possible without such a collaborative effort. So I'm very grateful to be presenting this on their behalf as well. And the second point I want to make is that I just want to take a moment to acknowledge the current climate we're in as scientists, which is affecting our science and basically all of us. So many of us in the US at the moment are, facing issues related to funding. We have students and researchers facing deportation simply by, standing up for what they believe in or voicing their opinions. This is not just true for US, it's also true for my home country. We have students who've been arrested for peacefully protesting and executing their, constitutional rights. So I just want to take a moment to acknowledge that and, to kind of voice what's happening around us. And I know it's not always possible to stand up for these things, but I think it's important that we take a moment and show support and think about it as much as we can. Anyway, that being said, I want to go back to science, which is always more fun to do. So as said, I will be talking about our recent paper, and this is on neutrophils and their unique reprogramming in skin and other barrier tissues. So 1 second. I don't know if the slides are moving forward. Okay. So what are neutrophils? Neutrophils are, very canonical immune cells. They are described as the most abundant leukocytes in human blood and the fastest responders to infection and inflammation. This is a very textbook kind of definition for them. So if you have any problem in your body, any infection or inflammation, neutrophils are going to be the first ones to go there and tackle that problem for you. What's also known about them is that they have quite a short half-life, which was kind of thought to prevent them from having adaptations into unique settings they are. So they were mainly always regarded as these cells that go and solve an existing problem through their canonical functions, such as phagocytosis, reactive oxygen species production, degranulation, and whatnot. But they were predominantly regarded in what we call as more immunological settings. But in recent years, we kind of realized that's not only the case, because neutrophils are not only existing in our tissues in immunological settings or in cases of problems, they also infiltrate our tissues at steady state. So they can be found in many different tissues, of course, with varying abundances. As you can see here in the spleen or lung, obviously in the bone marrow, they're fairly abundant, whereas in other tissues such as skin, muscle, their abundance is less. And what has been also shown that this infiltration into tissues follows a circadian pattern. This will become more relevant in further slides in my talk. But basically, if you take the skin, they seem to kind of infiltrate our tissues or skin tissue during the day. Their numbers peak around in the afternoon, and then they go down again. What has also recently been shown that neutrophils actually in our tissues have a half-life longer than what has been sort of thought of. Again, if you take the skin tissue, neutrophils in the skin have a half-life of about 18 hours. And this is quite striking because this allows them to actually adapt to changes in our tissues or whatever the cues they're receiving in our tissues, right? So this is outside of what has been thought about them, that they are not amenable to a lot of changes. So 18 hours of half-life allows for transcriptional changes, changes in protein levels, and general ability to regulate their environment. And this actually has been shown beautifully by Ivan and Andrea in this 2020 cell study. I think Ivan had already presented this before for Open Box Science. In that paper, they looked at the transcriptional programs that are activated in neutrophils in different tissues. And they specifically focused on the lung, where they showed that neutrophils in the lung sort of assume this angiogenic programming. And these cells localize around certain blood vessels that were positive for the chemokine CXL12. So neutrophils would localize around these blood vessels, and they were shown to help the tissue revascularize upon an injury. So this is already showing us that they are doing other functions than, just canonical immune responses. So this paper has been quite fundamental for me to kind of understand the capacity of neutrophils for plasticity and adaptation. It's also, I think, still, it's my favorite paper that I've read. And our study actually comes from kind of their finding and builds up on what they've seen. So when we started our project, we wanted to dive into this programming of neutrophils. And this is work of Tommaso and Jackson, who started this project before I was even in the lab. So, so quite a few of the data you'll see here is also their work. So what they did is that they compared neutrophils from barrier tissues, such as lung, gut, and the skin, to neutrophils from non-barrier tissues, blood, bone marrow, spleen. So these a little bit more sort of hematopoietic tissues, really like bone marrow and spleen, and then the blood, which is a very major component for neutrophils as well. And the lung, gut, and skin would be the barrier sites that our bodies kind of are exposed to certain external agents. And what they found is that neutrophils in barrier tissues had a specific transcriptional reprogramming for genes related to extracellular matrix production. So these cells expressed a variety of collagens, genes for collagens, laminins, and other ECM genes. They obviously also expressed matrix degradation enzyme-related genes, which are already known as an important part of their functionality. And they also express genes related to sort of compaction of matrix, such as enzymes that are relevant for matrix compaction and stabilization. So this is quite striking because this is definitely out of what we know them to do, right? And this was not just a transcriptional signature. We've also seen that these cells also express proteins of matrix. So again, comparing to a non-barrier neutrophil to a barrier neutrophil isolated from the lung, we see that neutrophils from the lung express collagens, laminin, fibronectin. And again, when we look at the kind of the abundance of neutrophils in different compartments, what we see is that the non-barrier tissues do not have any of these cells, whereas neutrophils from lung, gut, and skin, especially skin, have a very high fraction of these neutrophils that have an enhanced matrix signature. So this was quite striking, and this we validated further in different types of skin as well. So obviously not all skin in our bodies or the mouse body is the same. So we looked at different types of skin in mice, ear skin, back skin, plantar skin, which is like the palm, and the tail skin. And all of these different sites harbored these matrix-producing neutrophils. And not only mice, but also neutrophils from humans. I don't know why this slide look ugly, but also had collagen-producing neutrophils. So again, we compare it to non-barrier tissues, such as blood and spleen, And then we also looked at lung and skin. And again, you can see that skin neutrophils have the highest abundance for this matrix-producing neutrophils. And this we also saw in skin biopsies, not just in isolated cells. So here you have pointed with white arrows. I guess you'll have to take my word for it because it's a bit blurry. You have neutrophils that are negative for collagen in the skin. And then with the yellow arrows, you have neutrophils positive for collagen. And you also can, I guess, see the extracellular collagen that is red here. So this was quite a striking finding that neutrophils produce matrix proteins in mouse skin or mouse barrier tissues, and as well as in human skin. But then our question was, what do they do? Like, okay, they do this, but what's the relevance of this? So to understand that, we basically compared wild-type control animals with neutropenic animals. So these mice basically have a inducible diphtherotoxin receptor in the neutrophils. So when you give diphtherotoxin, neutrophils die off. So we can deplete them and we can investigate the differences in this neutropenic setting. And what we've seen is that if we look at the neutropenic mice, the organization of the fibrils, collagen fibrils, were significantly disrupted compared to the control mice. So this is the skin of a wild-type control animal, and you can see the fibrils are intact and quite well organized, whereas if you look at neutropenic animals, the structure is altered, and we also see that the width of fibers are significantly reduced. So this is very striking in my opinion, because first of all, this is a naive tissue, naive skin tissue, which does not harbor that many neutrophils to begin with. And you already have your fibroblasts, which are the main cell types that are responsible for organizing matrix in this tissue. But just by lacking neutrophils, the organization is significantly disrupted. And this was not only organization, but the mechanics of the tissue was also altered quite significantly in the skin. So we compared the stiffness of the skin tissue by using like a homemade device that kind of stretches the skin and understands how much force it can retain. And what we saw is that in the neutropenic mice, the stiffness of the tissue was again dramatically, significantly reduced compared to the wild-type counterparts. So absence of these cells alters the organization of the skin, and it also alters its stiffness, so mechanical properties, which was quite striking. The next thing we wanted to understand then was, well, I mentioned that neutrophils infiltrate tissues in a circadian manner, right? So we wanted to see if any of these properties would have a circadian regulation attached to it. So this was a very intense experiment that was done by Tommaso. And what he basically did was he measured the stiffness of tissue within a day at different time points to see if there were any circadian alterations to tissue stiffness. So there's no manipulation, just at different times of the day, he measures the stiffness of the tissue. And what we see is actually that at basal levels, the stiffness of the tissue itself changes throughout the day. So you can see in the evening, later in the day, at night actually, the stiffness of the skin is much more different than it is throughout the day. So it kind of also coincided with the infiltration of neutrophils in the skin, which happened in the afternoon and the evening, and the stiffness of the tissue increased thereafter. So basically the stiffness of the skin is also regulated in a circadian manner. So you can see here as well that the stiffness of skin is significantly higher at night compared to the day. But if you deplete neutrophils, so if you're again in a neutropenic setting, this alteration is lost. So basically, the circadian regulation of tissue stiffness is also regulated by neutrophils, which was again a very striking and interesting finding to kind of attribute to these cells. Well, we found out that they are doing a lot of stuff regarding the mechanics of our skin, but we then wanted to be able to understand what is actually behind this program, right? What are the mechanisms regulating this in a way to kind of find ways to modulate it a bit more specifically than just depleting the entire set of neutrophils. So for this, we did an in vivo screening assay where we basically use different treatment schemes or different conditions to understand if any of them would interfere with the matrix signature of neutrophils. So here, for example, on top, you have the control condition, and you have a certain expression of collagen, elastin, and laminin. And if any of the treatments or conditions would affect the signature, so everything is normalized, if there's an increase of enhancement of the matrix signature, you would see the color changing red. And if there's a decrease or loss of signature, you would see the color to go to the gray. And what we've seen is that by blocking TGF-beta, we significantly, very dramatically, basically altered this matrix program. So the program was abrogated in neutrophils by blocking of TGF-beta, which also wasn't very surprising because TGF-beta is a cytokine that is very important for fibroblasts. Their survival, function, development is all affected from TGF-beta. And again, as I said, fibroblasts are actually the canonical cells to produce and modulate matrix. So it makes sense that neutrophils with the specific program are also affected from similar signals. So once we found out that TGF-beta was one of the drivers of this program in neutrophils, we of course wanted to be able to modulate it. So what we did was to generate a mouse strain that lacked TGF-beta receptor specifically on neutrophils. So basically what we did is we crossed the neutrophil-specific Cree, MRP8, with a TGF-beta receptor flox mice. So these mice only have their neutrophils affected. And in theory, the TGF-beta signaling in the rest of the body should be same. And we, of course, validated this by checking the immune composition in peripheral blood and saw that it was the same as healthy controls. We've also validated that these neutrophils were immunocompetent in these animals because obviously TGF-beta is also an important immunological signal. But we saw that neutrophils were able to phagocytose the same as healthy, able to produce reactive oxygen species, and they were also able to migrate as efficiently as wild-type neutrophils. But what was different is that these neutrophils basically were deficient in the matrix program that we described before. So this mouse line then allowed us to investigate this specific reprogramming in neutrophils in a more detailed way. So we went back again to compare the structure of the control animals to these TGF mice. I'm going to call them TGF because it's just easier than saying neutrophil-specific TGF-beta receptor deletion. So basically, the TGF mice, again, had a disruption in their matrix organization. The width of these fibers were, again, smaller in the TGF mice compared to the wild-type animals, showing that, again, just by abrogating the specific program in neutrophils, you are able to impact the structure of an entire skin. We, again, did the mechanics analysis. So we measured the stiffness in the TGF mice and compared to wild type. And so again, the stiffness was significantly reduced. But on top of that, we also looked at the permeability of the tissue. So this is a very simple permeability assay. What we do is that we add a drop of Evans blue dye onto the skin, your skin here as an example. And as you can see in the wild type mice, this dye sort of permeates to a certain extent, but in a kind of a limited way. So probably like through micro wounds or hair follicles, it is able to enter the skin, but does not really spread much. Whereas in the TGF mice, you see that the dye spreads and permeates much more strongly in the skin. So not only the stiffness of the tissue is affected, but the permeability of the skin is also dramatically impacted when neutrophils lack this program. Of course, with these findings, we had to think around the idea of why neutrophils do this and why do we need them when we have a cell type like fibroblasts who are specialized to execute this function, right? Like they are, again, fibroblasts are known to do this. They are basically the main cells who are resident in our tissues and much more abundant than neutrophils. So why would we need neutrophils to take over such an important structural program? Not take over, but maybe assist such an fundamental structural program when we have fibroblasts. So we had to think around this idea quite a bit, and we had two main hypotheses. So both of them actually derive from the selective advantage of neutrophils, which is their high motility, right? Because fibroblasts are abundant, but they're stationary in our tissues, whereas neutrophils are much more motile, and they're, easy to respond to specific cues. And so what we thought was that either these cells could be targeting specific tissue areas or niches, or they could also be kind of employed to assist fast-paced dynamic responses. So then we went ahead and tried to test these hypotheses, basically. Just to kind of picture what I mean by, their dynamicity in a tissue, or how their motility can assist this function. So this is a 2-photon imaging of skin. This is a fibroblast reporter. This is a neutrophil reporter. And you can see already that the fibroblasts in the skin are quite abundant. And within the same kind of area, you have one little neutrophil, which is obviously way less abundant. But if you look at it on a time lapse, you see that your fibroblasts are just basically sitting there stationary, whereas this one little neutrophil is basically going around the entire tissue for you. And this is an extravascular neutrophil because you can actually see the vascular neutrophils shooting around here if you look a bit closer to the video. So we did kind of like a mathematical modeling to understand this. And what we kind of derived is that all of the fibroblasts in your skin covers about 16 to 20%, 20% max of your entire skin area. And they're, stationary. They're not going anywhere. But that neutrophil that was kind of moving around our tissue can span, in theory, the entire skin area within a day. And all of the neutrophils in the skin can span the entire skin area in less than an hour. So this is their potential. And this is why we think that they are kind of specialized to utilize this function and assist fibroblasts in a way when the assistance is needed. Another thing we looked at is, again, around the same idea of that they might be localizing into specific regions of the tissue. And so this is an entire image of a mouse ear skin. So you can see the cartilage running in between, and then you have epidermis on both sides. And what you see here is that there's like a subepidermal region, which is kind of denser in collagen structure compared to the lower dermal regions. And when we look at it closely, we saw that neutrophils were preferentially localized to the subepidermal region, and they were significantly less abundant in lower parts of the dermis. You do still find neutrophils in the lower parts of the dermis, especially those that, extravasate or so, that are closer to blood vessels. But we've seen a clear preference towards the subepidermal region of the skin. And we then went on to analyze kind of the collagen organization in these two parts of the skin, the subepidermal region versus the lower dermal region. I here show you an image. So this is transmission electron microscopy, which shows individual fibrils of collagen. This is from subepidermal region. And what you can see in these wild-type animals is that there are different sizes of collagen fibrils, right? There are tiny ones, relatively bigger in size, intermediate size ones, and there are the very large ones that are shown in yellow circles, which are, I'm guessing, higher than 0.2 micrometer in diameter. But there is a variety of different fibrils in the subepidermal region. And what we've noticed is that when we quantify this, very large fibrils are actually kind of almost unique to the subepidermal region, whereas in the lower parts of the dermis, you don't find these fibrils in the wild-type animals. When we looked at our TGF mice, what we see was quite striking. So this is again an image from a subepidermal region. And you can see that basically there are no large fibrils in these mice. Not in the subepidermal region, nor in the lower parts of the dermis. So what we thought then is that this was in line with the fact that we found these cells, our neutrophils, around subepidermal region. So we thought that it's possible that these cells are involved in the formation of these large fibers. They might be building them, they might help them become more compact. But in the absence of this programming in neutrophils, the body, the skin lacks these fibrils, which was, very striking. So we've shown so far that neutrophils are clearly affecting the organization and structure and mechanics of our skin. They express the specific program, transcriptional program, and they have the proteins for ECM. But what was more of a challenge in a way is to show that these cells are actually depositing collagens. So it is rather tricky to prove that an individual cell type is responsible for deposition of collagen in vivo, right? So we needed to come up with a strategy to show that these cells were depositing collagen in skin tissue in vivo. And to be able to do that, we collaborated with Dr. Yuval Rinkovich, and who helped us design this CNA35 reporter mouse line. So CNA35 is actually a native collagen binding protein. It's commonly used for staining of collagens. For instance, like it's conjugated to certain fluorophores, so people conjugate it to certain fluorophores and then use it for ex vivo staining of tissues to visualize collagens. So what we've done is that we kind of use that sequence to generate this flux mice. We've also conjugated it to like an mChar reporter to be able to visualize the protein in vivo. But what was important for this mouse line is that the sequence of the gene lacked an export domain, which meant that the CNA35 mChar protein could not really be exported out of the cell alone because it couldn't have been processed through ER and Golgi. So only when it's bound to collagens inside the cell, then it can be transported out of the cell. So this kind of allowed us to track the collagen, so the bind collagens inside the cell with the NAM cherry, and then track their deposition outside the cell and into the tissue. So using this flux mice, we crossed it to a neutrophil and fibroblast specific cleaves and analyzed the position of collagens by the two cell types in skin. And we were lucky that the model worked, because if you are experienced with generating mice, you know that they don't always work. But what we could see actually is that both the neutrophil reporter and the fibroblast reporter, we were able to track the deposition of collagen in the skin. And as you can see that the signal from the fibroblast reporter is much more stronger than the ones from the neutrophils, which is expected. And what was also really reassuring to confirm is that the position of collagen by neutrophils were relatively closer to epidermis than the deposition of collagen by fibroblasts. So here it's quantified, and you can see that neutrophil-derived collagen is closer to the epidermis, significantly closer to epidermis than the fibroblast-derived collagen. So it kind of validated our findings with transmission electron microscopy and match with the fact that we were finding these cells mainly around subepidermal regions. So we were able to show that these cells are able to deposit collagen. They're able to modulate properties of the skin, mechanical properties of the skin. And regarding their advantage for fibroblasts, again, we mentioned their motility and their ability to respond fast to stimulus. So in more of a dynamic setting, they would be relevant. So then what is the kind of the setting that neutrophils could help the body respond faster to a stimulus? And we wanted to understand basically how the program was utilized in a dynamic setting or an acute need of the body. And for that, we used a very simple wounding model. So we generated skin wounds on the ear of the mice with basically needle pricking. So these are tiny micro wounds, let's say. And then we kind of observed the kinetics of the development of the wound, recruitment of neutrophils, and how the collagen is deposited around the tissue. So looking at the kinetics, as expected, neutrophils are already there within the first day, fastest responders to infection and inflammation. They're going to be there. That we knew already. And then they kind of form this ring around the tissue or around the wound in the next few days. And as the wound closes, neutrophils are basically going away from the site. And what we've noticed as well is that these neutrophils that come in early day, so the ones that are there until day one, they do not have collagen inside them. So they don't have this matrix program. But those that have the matrix program and the collagens come a bit later into the tissue around day 2 and day 3. And this was quite striking because this coincided with a formation of a collagen ring around the wound. So as you can see on day 2, you have this collagen deposition happening around the wound, which continues to build around day 3 as well. And then it persists even after the neutrophils are gone. So it's a bit more stable than the presence of the cell itself. And if you take a closer look at this ring, what you also see is that it basically seals the wound from inside. So it kind of looks like it's providing some form of a protection to, close the wound, not in a healing way, but like creating a barrier around it. And when we compare this with the wild type and the TGF mice, what we see is that, so again, this is the edge of a wound, and you have your neutrophils here. And if we take a closer look, you can see that, so neutrophils stand for MRP14 here. These are positive for collagen in the wild type mice. And you can also see the extracellular collagen around the cells. But if you look at the TGF mice, you still have the cells. They are able to migrate to the wound just as well. You can see the counts are the same as well, but they do not have the collagen inside them. So they don't have the program active, and they don't have this extracellular deposition of collagen either. So it's a bit clearer here is that these TGF mice lack the deposition of matrix around the wound. So they lack this ability to seal the wound. So then we thought, what setting this could be relevant at? And obviously, one setting this would be very important is probably a secondary infection, right? Because wounds are sites where a lot of pathogens and can enter our body. So if you have an open wound, it's likely to get infected. So body has to have mechanism around it to protect. And we thought that this ring that is forming around day 2 or 3 can actually be a way for the body to kind of prevent or reduce possible secondary infections. So we employed, again, a very simple infection model, combined it with our wounding model. So we induced our micro wounds again as before, just the same. And then we waited 3 days until this matrix ring formed around the wound. So it again did that sealing action. And then we added a drop of bacteria onto the ear skin. We kept that bacteria, like the drop, for an hour, wiped it off, waited for another 3 hours. And in total of 4 hours, we basically terminated our experiments and then analyzed the skin. The reason why we chose such a short time point was to avoid any other confounding factors that are important for, an infection, right? So to avoid that, this was an action mediated by, differences in immune response, or if it was an action mediated by like significant bacterial proliferation or not. So we wanted this to be related to basically a physical hindrance of bacteria to spread in the tissue. And what we saw when we compared the wild type to the TGF mice is that the wild type mice, which had this ring around the wound, actually contained the bacteria. So this is a staining for Staph aureus, basically at the wound edge. So the bacteria itself could not have the possibilities to spread farther into the skin. Whereas in the TGF mice, which, as I showed you before, that don't have this ring or like sealing, the bacteria was able to spread much more around the tissue, so significantly higher distance from the wound edge. So this kind of showed us that while this program is quite important for our naive tissues, but it is also important in an immunological setting where we, already acknowledge that neutrophils contribute a lot, but not through their immunological function, but through a physical structural function as well. So there's more to these cells than what we acknowledge them for, basically. So that brings me to the end of my talk. I just want to give a brief summary of what I've been saying so far. So basically, we've shown that neutrophils contribute to the structural organization or framework of our barrier tissues. I've shown you data predominantly from the skin, but we see these cells in lung and gut tissue as well, to a fairly less extent, but we think they are likely to contribute to the organization there at different settings, of course. And we've also shown you that this matrix program in neutrophils depends on cell intrinsic TGF-beta signaling. So why I think this is important is that this gives us a way to be able to modulate this program, right? We can use it to our advantage or block it when it's necessary, because matrix deposition is relevant in various different conditions. And these neutrophils, so the matrix producing neutrophils, are really important for our naive tissues, naive barrier tissues, but they're also quite important in an immunological setting where they contain pathogens and prevent the spread of infection. So this is basically what we have kind of showed or seen in this paper. And currently what we are also exploring is that, because this is such a novel function or program, let's say, we're trying to understand how this could be relevant in different settings too. And currently we're investigating how this could be utilized in fibrotic pathologies, because neutrophils are already, I guess, acknowledged quite a bit in fibrosis for their immunological or inflammatory roles. But in the hallmark of fibrosis is the change in ECM or matrix deposition. And we're now investigating these cells, whether they're contributing to, for example, pulmonary fibrosis and the mechanics of pulmonary fibrosis rather than inflammation. And we're also looking at cancer too, because there are certain cancers which have very prominent ECM deposition. One of them is pancreatic adenocarcinoma, for instance, and this aberrant ECM deposition is often linked to poor immune infiltration and poor immunotherapy response. So having an idea on how these cells are affecting these conditions also can provide us with ideas on how we can modulate and think of therapeutic options, which we had not thought about before. So this is more of a translational aspect of it. And from the more basic science perspective, we're trying to understand how this work is divided between fibroblasts and neutrophils. We speculated on some aspects and kind of came out with some ideas on it and data on it. But we want to see also the basically dynamic between them, if they're interacting, what are the signals, and also at what part neutrophils are contributing to the kind of the matrix itself. Are they only depositing? Are they helping compaction? Are they affecting the matrix deposited by fibroblasts, and whether fibroblasts are affecting matrix deposited by neutrophils? So this is from a very basic science perspective of it as well. So with that, that I come to my acknowledgement slides. I want to thank everyone who's participated in this work. So for those of you